So what this looks like for this particular patient, they have in the DPYD gene, they have these, they're homozygous for these two genetic variants that are inherited as a haplotype. This would confer a protein activity of the DPD enzyme of 1. So it's a very direct relationship of measurement between variant interpretation and the enzyme activity here. Their phenotype, they would be an intermediate metabolizer. They'd have decreased enzyme activity and an increased risk of severe drug-induced toxicity for the fluoropyrimidine drugs. And the therapeutic recommendation would be that they should reduce, have a reduced dose of that drug before they start treatment. So we did this across almost 77,000 participants to translate their individual genotypes to their therapeutic recommendation. And for each gene-drug pair here, we see the resulting therapeutic recommendation. In blue, whether those participants would be recommended a reduced dose of the drug before treatment based on their genotype. And then in yellow, a small slither of participants where they would actually be recommended not to take these drugs at all because they're completely deficient in the enzyme and it would have a severe consequence for them. So we then applied this to the number of prescriptions for these drugs using the NHS England prescribing data across any indication. And we resulted in around 14,500 patients a year could actually potentially have their prescription optimized with a lower dose or using an alternative drug, looking at just these 14 genetic variants. So this potentially has a great potential to reduce the number of hospital admissions that we see every year. So 67% of hospital admissions are predicted to be due to adverse drug reactions. And this costs around £380 million a year to the NHS. We also wanted to show look into the data to see if we could find the pharmacogenetic association ourselves. So we had to pull out from the hospital incident record data, matching that with the participants, the ICD-10 codes, the phenotype codes, which are likely to be associated with drug reactions. Also, in the separate cancer registry, pull out data for these participants on which drugs they were taking and whether they were taking these pharmacogenetic associated drugs. But what was really nice was we could see when carrying out a PHEWAS analysis, we could see that we did see significant phenotypes associated with DPYD variants in patients treated with capecitabine. So here you see significance for hypovolemia, neutropenia, and poisoning by primary systemic agents through this PHEWAS analysis. So we were able to recapitulate the pharmacogenetic association using this real-world data. We then went on to implement this as part of the 100,000 Genomes project by developing the bioinformatics pipeline and including pharmacogenetic results as part of the genome analysis that were provided back to the NHS Genomic Medicine Service. And so this happened sort of through the lifespan of the programme. Around 7,000 participants were screened. And around 5% were positive for a DPYD variant. From surveying the GMCs, we found that these were likely to be clinically relevant. NHS England then commissioned DPYD genetic testing, and it's included in the genomic testing directory. So it's now part of implementation for the NHS. What I wanted to highlight, though, was that of those 14 variants that we looked at, we looked across the different ancestry proportions within the 100,000 genomes cohort. And as you can clearly see, I've highlighted in red that many of those variants are not at all found in non-European ancestry participants. And so there's a real need to address the lack of evidence for pharmacogenetic related variants in more diverse populations, so that we can ensure equity of care. So to move on to my second example, around really how we can use variant interpretation at scale to help make safer and more effective drugs. So this is my work at Open Targets, which is a partnership to transform drug discovery through the systematic identification and prioritization of targets. So together we bring the academic institutes EMBL EBI and the Sanger Institute with our currently 5 pharmaceutical company partners, GSK, Sanofi, Pfizer, Genentech and Merck MSD. And our approach is to systematically use evidence to help build a therapeutic hypothesis between targets and diseases. And we do this through generating data through our research program, but also by integrating the wealth of publicly available data that's out there. And we build upon this target disease evidence level. This helps build therapeutic hypothesis where you might think about how you'd want to modulate that target in order to treat the disease. And the foundation of our evidence is genetics. Why? We know it's very important for drugs to be successful. So a drug's 8 times more likely to succeed and come to the market if it has underlying Mendelian genetic evidence for the association between that gene and disease, and twice as likely if it has GWAS evidence. Our group have shown over the past 10 years, around 2/3 of approved drugs have underlying human genetic evidence to support that association. And then more recently, Matt Nelson's group published the drug mechanisms with genetic support are 2.6 greater chance probability of success if they've got that underlying genetic evidence to support them. However, what's exciting is actually that we're far from reaching peak genetic insights to aid the discovery of targets for more effective drugs. So GWAS is useful for developing, providing evidence for targets diseases, but actually it's really tricky to use evidence from a GWAS signal to find the underlying causal gene for a disease. And I think we've heard a lot about that in the talks this morning. But here you've got a particular, signal. How do you identify which is the causal gene here? Most disease-associated variants from GWAS are outside coding regions. The causal gene is not necessarily the gene closest to the associated variant. Sometimes these variants can affect multiple genes, and the lead variant is not necessarily the causal variant. So it's a very different scenario from the pharmacogenetic variants, where we could very easily go from, we know that we've got this strong evidence for the functional consequence of this variant. And here we're sort of, we don't know what might be the underlying causal gene here. And how do we do this at scale? So our approach has been to develop Open Targets Gentropy, which is a Python package for post-GWAS analysis. It's automated, it's modular, and it really helps address this challenge of the ever-expanding data that we're facing, and enables us to do complex statistical genetic analysis at speed and at scale. So we work very closely with the GWAS catalogue team, who help us harmonise summary statistics from GWAS studies. We feed in GWAS from the GWAS catalogue, FinGen and UK Biobank, and do our own fine mapping to provide GWAS credible sets. We then work very closely with the EQTL catalogue team, who provide us with molecular QTL credible sets. That's pQTLs, eQTLs, splice QTLs, and 2QTLs. We carry out co-localization across GWAS, and QTL GWAS. And that all feeds into our feature matrix for our locus to gene machine learning pipeline, which I'll explain in a second, but I just wanted to show you some numbers. I don't expect you to read all this, but this is just to show you that this is very much at scale. We now ingest, thanks to the GWAS catalog team, over 100,000 GWAS studies. And we provide, for example, over 2 million molecular QTL credible sets. So our locus to gene machine learning pipeline, the goal here was really to help systematically provide a predictive score for the most likely causal gene underlying GWAS association. I won't go into the model in detail, you can read more about it in the paper, but it currently includes 28 features across 4 different categories. So it includes looking at in silico pathogenicity prediction from VEP, distance from genes, molecular QTL co-localization, and credible set related features. We because this is a computational biology conference, I had to show a snapshot of our pipelines. So this is our pipelines where the entropy at the top is orchestrated with all our platform ETL, and it's all run together. It takes only about 3.5 hours to run for a full Open Targets platform release. Which is, I think, incredible, since as one of the boxes at the top, the co-localization, used to take us about 2 months to run. So we've massively streamlined this, and it's at scale and it's exciting. So this all goes into our Open Targets platform release, which is our open source resource. The results from the L2G in our last release resulted in over 500,000 gene disease associations, supported by almost 1.5 million pieces of evidence. So this is really at scale. Here's an example of our Alzheimer's disease page. So you can search for a disease, a gene of interest, variant, or a study, or a drug. And here we have the evidence from not only the L2G pipeline, but also combined with other genetic evidence such as gene burden and rare disease genetics from ClinVar and UniProt, along with evidence from somatic mutations, known drugs and clinical trial data, perturbation pathway analysis, expression, extraction from literature and animal models, all providing evidence for targets that you can see, or genes that you can see on the left-hand side. And these are weighted and scored, prioritized based on a harmonic sum that we use for each data source. But you can now adjust that weighting, so you can alter the prioritization of those targets. So here, the darker the circle, the higher the association score that we give, but you can do your own adjusting of the weights for the different data sources and which ones you want to include. You can now also upload your own list of genes and immediately see the evidence that there is for that association with this particular disease, and filter this based on pathway, go terms, that kind of thing, to start to play around with your list of genes. You can now dig into the immediate evidence underlying that association and look at interactors of your target of interest to see, is there more evidence for another protein that interacts with my target. So here's what the locus to gene predictions look like in the platform. It's a score, but we want to kind of help you look into the black box, as it were. And we now provide SHAP values, which is based on game theory, to explain the contribution of the individual features or groups of features that go towards that final locus to gene predictive score. You can start to build within the platform a therapeutic hypothesis. So here we can see there there's evidence, genetic evidence for loss of function this is a classic example of the PCSK9 gene, where loss of function variation results in lowered LDL cholesterol. It's protective. We can then click onto the Campbell evidence to see that actually inhibitors for this target are developed to treat the disease. So you can start to think about using genetic evidence and the direction of that mechanism to think about how you might treat the disease. And on our target attribute view, you can now also start to ask questions like, has this target ever been drugged before? Would a small molecule, a PROTAC or an antibody be suitable to treat this, as a modulator? Is there a suitable animal model or probe that I can go forward with validation studies? And what are the possible risks of modulating this? How safe might it be? So we have evaluations and scoring for you to be able to ask those kind of questions. So in the platform, we're starting to bring in common and rare disease trait associations together at scale to start to understand variant consequences and gene function. So here I've just shown across our variants, the distribution of the types of evidence for each variant and the source that's coming from. So in pink, we've got pharmacogenetic evidence. Green is coming from more rare disease clinical type resources like ClinVar and UniProt. And purple is our credible set evidence coming from our statistical genetics pipelines. But you can see, there is an overlap here in the middle of variants that have evidence for a phenotype or a trait or a disease coming from all of those different evidence sources, which I think is really interesting. And you can then view this on the variant pages. So we have functional context information for now around 6.5 million genetic variants, both common and rare. And the union of these evidence sources, as well as a summary of variant effect predictions, which you can see we sort of use this nice visual because everyone says something slightly different. And you can also view your variant in the context of the protein structure if it's a coding variant and play around. We give this we've shown the alpha missense pathogenicity ratings here in coloration. So the redder the areas are the more pathogenic regions in the protein. And you can start to build with these different evidence sources a picture of what your variant function might be, what it might be doing. So for example, this one is likely to be deleterious. It's a missense in ApoE with associations from syndromic and common cardiovascular disease, but also associated with increased response to statin treatment. So all this is open to you. It's all open source, both the software and the data. This is the platform URL, and you can query our data in numerous ways. We've got the nice web interface. We have a GraphQL API, or you can use Google BigQuery or download everything and play around with it in that way. We have over 72,000 public users now across the world. However, I think that's probably an underestimate because a lot of people, because the software is open source, have their own customized versions of the platform internally that we're unable to know how many that is. And we have 3 million downloads a year of our data. And we'd love you to get involved. We have a hackathon in October. This is to celebrate 10 years of the Open Targets platform and an opportunity to get the community together to develop data analyses, new tools for target discovery. We've got the project call is open now, so please submit your ideas and register for the hackathon. It's hybrids, but we'd love to see you on campus. So to move on, and I don't know how much time I've got because of the interruptions, but I'll speed through this. To some of the challenges that we're all facing now that we've heard about this morning, and whether AI can help address some of these for us. So how do we harness the huge amounts of hidden knowledge and information in our growing data corpuses and help link that knowledge together? How do we integrate and interpret the ever-expanding, ever more granular, ever more context-specific data that's being generated? How do we provide this holistic view of the effect of a variant or a perturbation across different biological layers from the genome all the way to the patient? And how do we overcome the scarcity of patient numbers and multiomics data available to better understand currently untreated diseases? And we've got a range of projects across our Open Targets research programme and the platform that touch upon being able to extract knowledge from clinical trial data, the scientific literature, from drug labels. And we have a project in Sebastian, actually, I don't know if he's in the room, but he's around at the conference, to build representation of that knowledge through knowledge graphs and LLMs for conversational querying, building on the existing BioCipher and BioChatter frameworks. So go find Sebastian if you can to talk to him more about that. And actually, in the platform team, we've implemented the Google Croissant framework to enable our data to be a lot more interpretable by AI. And I have several projects that I'm collaborating with EBI colleagues on to harness AI in different ways. The first with Thomas Fitzgerald, Ewan Burney and Moritz Gustung around a pharmacogenetics project, actually. So expanding upon the model that they've, the Delphi model that they've developed to include genetics and drug exposure so that we can look at the relationship between genetics, drug exposure and phenotyping at scale across time. And then I also have a project with colleagues at EBI funded by the Chan Zuckerberg Initiative for the development of digital twins for rare disease. So this will combine mechanistic and machine learning Gen AI models to hopefully gain insights into what's going on in disease dysfunction in rare disease patients using multiomics data. And AI needs standardized, nice, harmonized, collated data. And I wanted to introduce the MVP launch of the Perturbation Catalogue, which will happen on Wednesday, which is very exciting. And this is led by Mallory Freeberg at EBI, and she's around, so go talk to her. We're wanting feedback here. So this is an Open Targets funded project, but it will be an EBI resource to collate data around everything perturbation. So at the gene level, at the variant level and expression level, and working on the metadata and standards that needed for that as well. And we have Stefan, who will be presenting a poster at one of our Open Targets projects. So go check his poster out on single cell perturb-seq analysis on Wednesday, I think. So hopefully I've covered how we can use variant interpretation at scale to help make existing drugs safer and more effective, but also develop new, safer and more effective drugs for patients. And to do this, we need to come together. We need to bring industry and academic academia together. We need to bring data together in order to do this. I had to include the Beatles because obviously we're in Liverpool today. But a big thank you to my team at Open Targets, all our partners, our collaborators, and our funders. Thank you very much. And I will just mention that I'm on this industry academic panel on Wednesday, so come along to that. But there's over 66 people from EBI here at the conference doing various talks, posters, and we have an exhibition stand to go check out. So thank you.
Thanks, Helen. Wonderful. Questions?
That's a great talk. So I have a question for that 14 genetic variant.
Oh, sorry, thank you.
Firstly is that maybe I missed that. How you identify that 14 variant from GOS? Then the second is that I show you have one slide that shows it has like a different frequency in the different populations. Like East Asian is related low. So I want to know, like, if you want to apply this to the different population, how do you consider the population stratifications? Maybe try different variant panels.
Yeah, absolutely. So it was from whole genome sequencing analysis, and we chose those variants. They were curated from evidence having, known association with the drug toxicity. So they were sort of very carefully selected that they would have clinical utility, and that was then analysed as part of the from the whole genome sequencing in the 100,000 genomes cohort. And yes, I fully agree that sadly, because of these 14 variants have generally been identified with an association, generally in European populations, for the association with drug toxicity, we need to study other more diverse populations in order to identify variants that are associated that have been missed in Europeans because they're a low frequency or so we're definitely the known evidence is definitely biased towards European data at the moment, which is a problem in genomics that we need to address.
Fabulous talk. So the GWAS analysis and a lot of the analysis you talked about has been applied to common disease, right? We do variant interpretation and drug discovery based on genetics for common disease, but there's a separate group of effort on rare disease. I'm wondering, what's your view on variant interpretation, the role of variant interpretation applying to common disease versus rare disease? Thank you.
Yeah, well, as part of the 100,000 Genomes project, we did diagnose, we had a diagnostic pipeline for rare disease patients, and the bioinformatics pipeline there was based around gathering evidence for genes to help with the variant filtering, and then identifying, likely loss of function, for example, variants within those green genes from PanLab. And then those would be sent back for clinicians to then evaluate using the ISMB guidelines, for example, in order to, truly say, okay, does this variant disease association and the existing evidence match my patient's phenotype, and is this like the likely causal variant? I hope that answers your question, but it is very, very different world from the common disease genetics and identifying associations there, and sort of that's how the worlds have collided, I guess, in the platform which is really nice to see that we're using evidence from both sides of those worlds.
We can get a last question while Thomas, you set up your talk.
I have a question actually to the gene trophy package, because as far as I understood, it's for GWAS variants defined or designed. But I unfortunately only have a SPEEDY database or SPEEDY identifier. Is it still usable for finding the gene you would expect to have an effect on, or?
For the Gentropy package,
Yeah.
you should be able. It's open source, so you should be able to run it.
Okay, so it's
And if you've got any, questions, the community page on the Open Targets website is the best place to ask any queries. But it should be deployable, and you should be able to run it.
nice. Thanks.
Thank you. Thanks again, Ellen.